So now we shall see what are the different types of inhibition. So enzyme inhibition can be broadly categorized into two types: irreversible inhibition and reversible inhibitions, as their name suggests. Irreversible inhibition is the inhibition where the enzyme is permanently inhibited. So we cannot remove the inhibitor from the enzyme, and hence that enzyme which is inhibited by a irreversible inhibitor is no more useful. But in case of reversible inhibition, the inhibitor can be removed from the enzyme by different processes such as dialysis, etc., so that we can regain the activity of the enzyme. So this irreversible inhibition can be divided into two categories based on their mechanism of action. So one type of irreversible inhibition is non-suicidal. So non-suicidal inhibitions are the amino acid modifiers, or where the inhibitor binds to the enzyme and permanently modifies the enzyme. So non-suicidal inhibitors directly bind to the enzyme, either to the active site or any other site, but they leave the enzyme changed in their structure so that the substrate can no more bind to the enzyme and the product is no more formed. So for example, diisopropyl fluorophosphate is one of the inhibitors of acetylcholinesterase, one of the very important protein involved in the neural signaling processes, and this inhibitor, which is used as a nerve poison, permanently damages the protein by covalently modifying it. Similarly, aspirin, one of the drugs which is used to inhibit cyclooxygenase enzyme, acts by covalently modifying the enzyme. So similarly, there is another set of inhibitors which are suicidal inhibitors. So suicidal inhibitors, in the sense, here the inhibitor binds to the enzyme but doesn't directly inactivate them. Rather, they lead to the formation of a reactive intermediate which in turn bind to the enzyme, and that binding is much more stronger than the binding of the initial substrate. So here the enzymes produce the intermediate, thereby permanently inhibiting themselves. So in this way, enzymes kill themselves. Therefore, they are termed as suicidal inhibitors. For example, penicillin. So penicillin is an antibacterial and it inhibits the enzyme transpeptidase. So transpeptidase is the enzyme which is involved in the cell wall synthesis of bacteria, and penicillin suicidally inhibits this enzyme. Similarly, monoamine oxidases, which are involved in production of neurotransmitters such as serotonin and dopamine, are inhibited by an inhibitor which is N,N-dimethyl propargylamine. So this inhibitor is a suicidal inhibitor for this enzyme, which permanently damages the enzyme and inhibits synthesis of these neurotransmitters. So they are very important for the medicinal point of view, which are used as drugs. So now coming to the second category of inhibitors, which are reversible. So reversible inhibitors can be categorized into four different types. So they are competitive inhibitors. So competitive inhibitors are the inhibitors which compete with the substrate for the active site of the enzyme. So they bind to the enzyme, but they compete with the substrate. So they are in part or in whole analogous to the substrate in their structure. so that the active site doesn't distinguish which is the substrate and which is the inhibitor, and this leads to inhibition of enzyme. So the second category of reversible inhibition is non-competitive inhibition. So in non-competitive inhibitions, the inhibitor binds to the enzyme but doesn't compete for the active site. And one more important thing is to remember that non-competitive inhibitors bind to the enzyme substrate complex rather than the free enzyme. So the third type of inhibitors are mixed, where the inhibitor binds to both the enzyme and the enzyme substrate complex. And here the whatever changes we see in the kinetics and the properties is a mixture of both competitive and non-competitive inhibitors. And the fourth type of inhibitors are called non-competitive inhibitors. So here it may be noted that non-competitive inhibition and on-competitive inhibitions are not synonymous. On-competitive inhibitions happen when the inhibitor binds to both enzyme and enzyme substrate complex with equal affinity. So this is a rare phenomenon which sometimes happens, but this should not be confused with the non-competitive inhibition. So rather it is a mixed inhibition where the inhibitor binds to both enzyme substrate complex as well as the enzyme. So now we shall see what changes happen, or how should we characterize or distinguish different type of inhibitions.